So here's a DNA sequence, and we can put in the polarity notation. So 5' on the left, 3' on the right. And the question was, if there was a restriction enzyme that digested this double helix, what happens to polarity labeling when you get two new ends, essentially? So I'm going to write in the second strand. Using base pair complementation. And that's the opposite polarity. So we've got anti-parallel strands. And then the question is, where is the restriction enzyme site in that sequence? So there's a palindrome there. AAGCTT. And that's the restriction site for the enzyme Hindi3, which I just had to look up. And Hindi3 cuts the nucleotide backbones at the places where I'm just drawing these little carrots. So it'll cut between those two A's on each strand. And the important thing about notation and polarity is to remember, that every nucleotide has a 3' and a 5' end. So if I just drew a T, it has a 5' carbon and it has a 3' carbon. So even a single nucleotide has polarity. If we looked at that same sequence after it was cut by Hindi3, then you would have 5' TGCA. And that's where the cut occurs. And we'd have the rest of that top strand. AGCTTCAC, that 3' end. So there'd be a gap there where Hindi3 had cut on the top strand. And likewise, the bottom strand would be ACGTT. So there's no space there because that strand's not broken. CGA. And then there would be a gap where the enzyme had cut the other strand. So there's a gap there, and there's a gap there. These are still hydrogen bonded to each other. But if you heated this up, what would happen? If you denatured these chromosomes, in other words, then you'd have, I'm just not going to write out the sequences, but I'm just going to write out according to the lengths of the backbones. You'd have something that looks like that, and it would be base paired to something that isn't base paired in that part of.
It doesn't cut the base pairs thing. I imagine it cut like here, like to the point where like these weren't connected.
That's
to these
correct.
anymore.
So if we heated them up, then this is what we would have.
Oh, okay.
Or if we heated them up and cooled them back down, so that the double-stranded parts that could form did form, then you'd have 1, 2, 3, 4 nucleotides that aren't base paired with each other. So that's what that double helix would look like after the cut. Is those 4 nucleotides here in the middle that are in between the two backbone breaks are single-stranded at this point. So you could add DNA ligase and ligate the backbones back together, that enzyme that finishes DNA replication by tying the backbones together, phosphodiester backbone. But this is what this would look like. And these are what we call, although we haven't talked about this in class, these are sticky ends. Yeah, they're complementary to each other, and that's what those would look like. Now, your question specifically was about polarity. So this was the original polarity of the ends of the original molecule of DNA. So the question is, what happened here in the backbone when we broke between those two A's? Well, there's a hydroxyl there. There's a hydroxyl here. Let me choose a different color. That was covalently linked to a phosphate in the backbone. That's the 3' hydroxyl and the 5' phosphate. So that's what happens when a restriction enzyme breaks the backbone, is it separates the OH from the phosphate group on the backbone, so they look kind of like individual nucleotides again. Which means that that is the polarity of those ends, P, 5'. So in other words, what was a continuous piece of DNA, 5' to 3', becomes two discontinuous pieces, but they always have the same polarity orientation. So just by cutting, we've separated the 3' hydroxyl from the 5' phosphate, so we could label those as ends of those strands. And likewise, the opposite strand has the opposite polarity.